Hello, fellow learners and science enthusiasts. Welcome to our exciting YouTube lecture today, where we're diving deep into the fascinating world of the animal tissue and the intricate web that holds it together, the extracellular matrix. I am Steven, and this is the Cell and Molecular Biology lecture series. I am your lecturer, and together we're about to unravel the secrets of this crucial biological structure that underpins the functionality of living organisms. The title of this lecture is The CellVerse Lining, where cells meet their world. In this particular lecture, we will identify the structures and functions of your ECM in animal tissue, relate the potential applications of organoids in research and clinical settings, describe how a cell surface protein can be involved in both cell adhesion and transmembrane signal transduction. Explain the functions of selectins, immunoglobulins, integrins, cadherins, adherins junctions, and desmosomes. Explain how the structure of a tight junction contributes to its function. Distinguish the mechanisms of a short-range, mid-range, and long-range intercellular communication, also describing the function of each component that makes up the plant cell wall. Explaining lastly how some characteristics of cell walls may have facilitated the evolutionary transition from water to land. But before that, I will just like to present this figure um, created with biorender of this research entitled The Extracellular Matrix and the Immune System, which is a mutually dependent relationship. In letter A, it will show you the physical and molecular properties of the ECM that controls immune cell positioning and the migration within a tissue during disease or the pathology. In letter B, this is aging not only that alters the mechanical properties of your ECM, which are sensed by the immune system, but also reduces your mechanosensors, those receptors that senses the movement that downregulate pro-inflammatory pathways. In C and D, this is the immunomodulatory cytokine IL-13 that can remodel the mucous barrier, whereas in letter D, this is immune cells themselves that can carry the matrix across the tissues to help build an ECM scaffold for repair. We start with the overview of the extracellular interactions. We have the cell organization. Materials that are present outside of your cell membrane plays an important role in the life of the cell. Most of the cells in the multicellular plant or animals are organized into clearly defined tissues. This is all about the hierarchy of biological organization. We remember that atoms make up your molecules, molecules make up your organelles make up your cells, and cells make up the tissues make up the organ. And there are a lot of diverse activities that are being regulated by this. Cells interact with their environment, extracellular environment for instance. In this particular figure, you will show you the overview of how cells are organized into tissues and how they interact with one another with their extracellular environment. In this schematic diagram of a section of a human skin, the cells of the epidermis are seen to adhere to one another by specialized contacts. The basal layer of epidermal cells also adheres to an underlying non-cellular layer called the basement membrane. You can also see your dermis that is consisting largely of extracellular elements that interact with each other and with the surfaces of scattered cells, primarily fibroblasts. Your cells contain receptors that interact with the extracellular materials and transmit signals going into the cell's interior. Just to give you an insight and our background about your skin, your epidermis has a closely packed cells of epithelial tissue. Your dermis is a type of connective tissue, and these fibroblasts of the dermis may have receptors that mediate interactions and transmit the message. Let's talk about the glycocalyx. These are carbohydrate projections that form part of this glycocalyx on the outer surface of your plasma membrane. The roles of your glycocalyx is to mediate cell-to-cell and cell-substratum interactions. They can also become mechanical projection, uh, barrier to molecular movements, and there are regulatory factor binding sites. This is in this first figure on the top is the basal surface of an ectodermal cell of an early chick embryo. You see here that there are two distinct structures which are closely applied to the outer cell surface, and they can be distinguished. The first is an inner glycocalyx and an outer basement membrane, or the BM. And letter B below on the left is an electron micrograph of the endothelial glycocalyx in the coronary capillary, showing you an extensive, okay, this is different from the first figure. The glycocalyx here is showing an extensive figure or shapes. And letter C is the molecular model of the glycocalyx, which are showing you some legend. The sugar chains are shown in green, and they are being bound to a transmembrane protein which is colored in red. One of the best defined extracellular matrix, or the ECM, is the basement membrane that surrounds the muscles, the nerves, and fat cells. Underlies the epidermis of the skin, the digestive and respiratory tract, and the lining of your blood vessels. This is the basement membrane, or also known as the basal lamina. In the figure on your left, A is the scanning electron micrograph of human skin. It shows you also the epidermis has been pulled away from part of the basement membrane, which is visible. You can see beneath the epidermal cells. In letter B, this is a dense network of collagen of the basement membrane, which is revealed by scanning electron microscope after an enzymatic separation of the epidermis. Showing you in the last figure is the much looser collagen network of the dermis is being visible here, with an adherent fibroblasts labeled with an asterisk. I don't know if you can see the asterisk there, but once you see the asterisk, these are the adherent fibroblasts. Macromolecular organization of the ECM is one of the purpose of this lecture, because they can take in diverse form in different tissues and organisms. The most of the proteins inside of these cells are globular in shape. These proteins are secreted into the outer portion, or the extracellular space. In the figure, you can see the proteins and polysaccharides, which is shown in this figure, and it will be discussed in the following sections. The proteins depicted here are the fibronectin, your collagen, your laminin, and they contain binding sites for one another, as well as binding sites for the receptors, such as integrins that are located on the cell surface. The proteoglycans are huge protein polysaccharide complexes that occupy much of the volume of this extracellular space. So you'll see here this blue bilayer, or the phospholipid bilayer, is the cell membrane, and the yellow is the cytoplasmic, the cytosol, and the cytoplasmic volume. Now we move forward with the components of the ECM. First is the collagen. Collagen molecule is the triple helix of composed of triple helix of 3 helical alpha chains. Collagens comprise a family of fibrous glycoprotein present in the ECM, and its most abundant protein in the human body. It's produced by fibroblasts, smooth muscles, and epithelial cells. There are 28 fiber types, and they are often mixed in ECM. This is the structure of the collagen. This figure depicts several levels of organization of a fibrillar collagen. The collagen molecule, or the monomer, is a triple helix composed of 3 helical alpha chains. Some types of collagen contain 3 identical alpha chains, and thus we call them homotrimers, while others are heterotrimers containing just 2 or 3 different chains. Each collagen 1 molecule is around 295 nanometer in length. In letter B, it will show you the collagen 1 molecules, which are become aligned in rows, in which the molecules in one row are staggered relative to those in the neighboring row. A bundle of your collagen 1 molecules, such as that's shown here, is forming you a collagen fibril. So I remember the staggered arrangement of the molecules that are producing the bands. You see here some of the horizontal black lines in this illustration across the fibril. They repeat with a periodicity of like 67 nanometer, which is equal to the length of the gap between molecules plus the overlap. Your collagen provides the insoluble framework that determines the many of the mechanical properties of the matrix. Showing you here the corneal stroma, which consists largely of layers of collagen fibers of that uniform diameter and spacing. So they are evenly distributed. The molecules of these alternate layers are arranged at right angles to one another, resembling the structure of a plywood. Tissue properties can often be correlated with the 3D organization of its collagen, such as your tendons and your cornea. Next is your proteoglycans. This is showing you a figure of a cartilage type proteoglycan complex. This is a schematic, letter A, showing you a schematic representation of a single proteoglycan consisting of a core protein to which a large number of glycosaminoglycan chains, such as GAGs, which are shown in red, are being attached. A proteoglycan from cartilage matrix, such as your aggrecan, may contain about 30 keratin sulfate and 100 chondroitin sulfate chains. Proteoglycans are found in the basement membranes, such as your perlecan and agrin, and they have only few GAG chains attached to the core protein. And letter B, these are the structures of the repeating disaccharides that make up each of the GAGs shown in this figure. All GAGs bear a large number of negative charges, and they are indicated by the blue shading. In letter C, in the cartilage matrix, individual proteoglycans are linked to a non-sulfated GAG called the hyaluronic acid or hyaluronin to form a giant complex with a molecular mass of about 3 million daltons. The box indicates one of the proteoglycans of the type shown in the first part, with the part A. And these proteoglycans have a repeating disaccharide structure, AB, where A and B represent two different sugars. They're common in basement membranes. They attract water and they're forming gel. Next is your fibronectin, showing you the figure of a structure of a fibronectin and its importance during embryonic development. A human fibronectin molecule consists of like two similar but non-identical polypeptides joined by a pair of disulfide bonds located near the C terminus or the C termini. Each polypeptide is composed of a linear series of distinct molecules which are organized into several larger functional units, illustrated by the colored cylinders in this figure. There are orange, you have lavender, you have yellow, you have blue, you have red, and yellow. These are repeating structures or cylinders. Each of these functional units contains one or more binding sites for either specific component of the ECM or for the surface of cells. Some of these binding activities are indicated by the labels. The cell binding site of these polypeptide that contains the sequence arginine, glycine, and asparagine, or the RGD as indicated. As discussed later in this lecture, the sequence binds specifically to a particular class of an integral plasma membrane proteins, we call them integrins, that are being involved in cell attachment and signal transduction. Showing you the inset, okay, on top of this image, 2 of the 30 or so repeating FN molecules that comprise the polypeptide. Your RGD sequence forms a loop of the polypeptide protruding from one module. Okay, the fibronectin consists of a linear array of 30 FN domains that give a modular construction. Affine type domains are found in blood clotting factors and receptors. They can bind to a lot of, again, myriad of ECM components, and they can bind to cell surface receptors. They have also, again, as I've said, they have important roles during embryonic development, and development is characterized by waves of migration. So, showing you a figure of a photo of this particular chick, okay, the role of cell migration during embryonic development. We have here a summary of some of the cellular traffic occurring during mammalian development. These are most extensive movements that are conducted by the neural crests cells that are shown in blue. They migrate out of the neural plate in the dorsal midline of the embryo, and they give rise to all the pigment cells of your skin, which is denoted in P. The sympathetic ganglia, or the SPG, the adrenal medulla, or the ADM, and the cartilage of the embryonics called MX, MD for maxillary and mandibular arches. The primordial germ cells, or the PGCs, are migrating from the yolk sac to the site of the gonads, or letter G, that are formed within the embryo. We have also the progenitors of the lymphoid cells that are transported to the liver, the L, the bone marrow, the BM, the thymus, the THY, your lymph nodes, your LN, and the spleen. Note that the pathways shown here connect the original sites of cells with their destinations. They do not accurately depict the actual routes of the cells. There are several organs, such as salivary gland, that are formed by a process of branching or cleft formation. Cell adhesion and shape determination also are fibronectin dependent. The structure here of the fibronectin and its importance during the embryonic development. We have here a human fibronectin molecule, which is consisting of two similar but non-identical polypeptides joined by a pair of your disulfide bonds located at the C termini. And you also can see here some of this micrograph where the neural crest cells are migrating out from a portion of the developing cheek nervous system. So they are being placed on a glass culture dish that are containing strips of fibronectin coated surface with alternating strips of the bare glass. The boundary of the fibronectin coated region is indicated by the white lines. It's evident that the cells remain exclusively on the fibronectin coated surface. Cells that reach the glass or the substratum, or showing here the arrowheads, tend to round up and lose their migratory capabilities. The arrow indicates the direction of migration. So they're also showing here the role of fibronectin in the formation of the embryonic salivary gland. Next is the laminin. So this is consisting of a family of at least 15 extracellular glycoproteins. Laminin has 3 polypeptide chains linked by disulfide bonds. They can greatly influence a cell's potential for migration, growth, and differentiation. They have also role in development of neuronal outgrowth, and they strengthen the basement membrane. So this is the figure of a laminin in this slide. What are the dynamic properties of the extracellular matrix? ECM can exhibit dynamic properties both in space and time. Spatially, the ECM fibrils can stretch several times their normal length. Matrix metalloproteinases degrade the ECM materials. The physiological roles of your MMPs, or your matrix metalloproteinases, are thought to be involved in tissue remodeling, embryonic cell migration, wound healing, and the formation of your blood vessels, or your angiogenesis. Abnormal expression of the MMPs are linked to a variety of diseases, including cancer. The engineering linkage here is the organoids. There are several different methods that support the growth of 3D cell cultures, and the engineering of organoids has made advances over the years. such as intestine, pancreas, stomach, and your optic op. They have also brain applications, such as disease research and pharmaceutical research. Showing you here a fluorescence microscopy image of the brain organoid. Neurons are labeled in green, and they are enriched in the outermost layer of the cortex, while neural stem cells are labeled in pink. Now we move forward with the interactions of the cells with their extracellular materials, first of which is the integrin. Integrin is a family of membrane proteins unique to animals. They are composed of two membranes spanning polypeptide chains, an alpha chain and a beta chain. The bent conformation of integrin corresponds to its inactive state. Once it is bent, it's inactive. Integrins with a bound ligand are found in an upright conformation. Showing you here the transmembrane domains of the two subunits which are in close proximity. Extracellular matrix and their ribbon drawings of their extracellular domains of an integrin in the bent. When say bent, it's inactive. When it's upright, it's active conformation. Changes are driven by the divalent metallic ions. Integrins have two major activities. First is adhesion of cells to their substratum or to other cells, and transmission of signals between the external environment and the cell's interior. There are also outside-in signals that can induce a conformational change in talin. Cytoplasmic protein kinases such as FAK and SRCs can be activated to phosphorylate other proteins. There are also outside-in signals transmitted by integrins can influence differentiation. We have motility, growth, and cell survival. Most of your malignant cells are capable of growing in liquid suspension, and normal cells can only grow and divide if they are cultured in a solid substratum. So these malignant cells or the tumor cells can grow in liquid suspensions. Next is focal adhesions. Focal adhesions are sites where cells adhere to their substratum and transmit signals in both directions across the plasma membrane. These cultured cells have been stained with fluorescent antibodies to reveal the locations of actin filaments. They are colored in gray-green and the integrins in red. The integrins are located in small patches that corresponds to the sites of your focal adhesions. Showing you below the cytoplasmic surface of a focal adhesion of a cultured amphibian cell, which is shown here after the inner surface of the membrane was processed for quick freeze deep etch analysis. There are also bundles of microfilaments that are seen to be associated with the inner surface of the membrane in the region of your focal adhesion. Focal adhesions again play a key role in cell locomotion, and they are dynamic structures. Next is hemidesmosomes. Hemidesmosomes are differentiated sites at the basal surface of epithelial cells where the cells are attached to the underlying basement membrane. It showing you here the electron micrograph of several hemidesmosomes showing the dense plaque on the inner surface of the plasma membrane and the intermediate filaments projecting into the cytoplasm. On the figure on the right is a schematic diagram showing you the major components of a hemidesmosome connecting the epidermis to the underlying dermis. We have here the alpha 6 beta 4 integrin molecules of the epidermal cells that are linked to a cytoplasmic intermediate filaments by a protein called plectin that is present in the dark staining plaque and to the basement membrane by anchoring filaments of a particular type of laminin A. A second transmembrane protein which is in BP180, if you can zoom it in, you can see here that is also present in hemidesmosomes. The collagen fibers are part of the underlying dermis. This is an experimental demonstration of the cell-to-cell recognition. Organs have a complex architecture involving a variety of different cell types, dependent on selective interactions between cells of the same type as well as between the cells of different types. Experiments demonstrated that separated cells would redistribute themselves so that each cell adhered only to the cells of the same type. Okay? When cells from different parts of an embryo are dissociated and then intermixed, your cells initially aggregate and then sort out by associating with other cells of the same type. These are the results of two such classic experiments shown in this figure. In letter A, show here in this experiment, there are two regions of an early amphibian embryo, the ectoderm and the mesoderm. They were dissociated into single cells and combined. At first, the cells formed a mixed aggregate, but eventually they sort out. The ectodermal cells, shown in red, move to the outer surface of the aggregate, which is where they would be located in the embryo, and the mesodermal cells, shown in purple, move to the interior, the position they would occupy in the embryo. Both types of cells then differentiate into the types of structures they would normally give rise to. So, this is an experimental, again, demonstration of your cell-to-cell recognition. Next is the selectins. Selectins are a family of membrane glycoproteins that bind to a specific oligosaccharides. Lectin is a term for a compound that binds to a specific carbohydrate groups. Selectins have a small cytoplasmic segment, a single membrane spanning domain, and a large extracellular portion. Here's all three types of the known selectins recognize and bind to a similar carbohydrate ligands at the ends of an oligosaccharide chains of your glycoproteins. In this inset, okay, the detailed structure of the carbohydrate ligand is being shown. The terminal fucose and sialic acid, these moieties are particularly important in selectin recognition. And the N-acetylglucosamine moiety is often sulfated. What is the role of cell adhesion in inflammation and metastasis? In the human perspective, inflammation is one of the primary responses to infection, but can produce adverse side effects like fever, swelling, redness, and pain. So these are the cardinal signs of fever or of inflammation. Following a puncture wound to the skin, white blood cells, the leukocytes from the bloodstream, are stimulated to traverse the endothelial layer that lines the smallest veins or the venules, and they can enter the tissue. Leukocyte recruitment has focused on three types of cell adhesion. These molecules are the selectins, integrins, and the immunoglobulin SF proteins. The endothelial cells that line the venules become more adhesive to circulating neutrophils through the display of P and E selectins. There's also what we call as your platelet activating factor, which is on endothelial cells, stimulating an increase in the integrin binding activity on the neutrophils. Bound neutrophils then change their shape and squeeze between adjacent endothelial cells into the damaged tissue. This figure is showing you the steps in the movement of neutrophils from the bloodstream during an inflammation. Okay? Cancer cells also escape these normal growth control mechanisms, and again they proliferate in an unregulated manner. Most malignant tumors are not readily contained. We have metastatic cells that are thought to have special cell surface properties. They can adhere elsewhere that are not shared by most other cells in the tumor. So, showing you the previous figure, these are like the steps of leading to the metastatic spread of an epithelial cancer or the carcinoma. So there we have here the fraction of the cells of the primary tumor which is losing their adhesiveness, to other tumor cells, and they gain the capability to penetrate the basement membrane. This barrier, the BM barrier, which is underlying the epithelial tissue, was being breached. Okay, these cells, which have assumed a mesenchymal-like appearance, they migrate through the surrounding stromal tissue. They cross the basement membrane of a blood or the lymph vessel, and thereby entering the general circulation. Then your cells are carried to other tissues, where they migrate back across the basement membrane of the vessel and enter a tissue in which they possess the potential to form what we call as the secondary tumors. Only a very small percentage of tumor cells that are released from the primary tumor manage to overcome these numerous hurdles. We have immune system also, which is being breached by the cancer cells. But they don't pose a threat to the life of the host. These circulating tumor cells have been isolated from a blood sample of a patient with a prostate cancer. This is an experiment. Even though the blood of a cancer patient may contain less than one cancer cell for every billion normal cells, these rare cancer cells can be selectively trapped on a chip that has been coated with antibody molecules directed against a cell surface proteins. In this case, they've used EPCAM, which is present on the cancer cells and absent from the normal blood cells. So we move forward with the interactions of the cells with other cells. Now, this is the Immunoglobulin superfamily. The human genome contains 765 distinct immunoglobulin domains, and these are the members of your immunoglobulin superfamily. Immune functions for most. Original function is likely cell adhesion mediators. They are also calcium independent, and they have developmental roles in neuronal growth and circuitry. Showing you here the L1 molecules. L1 is the member of the immunoglobulin Ig superfamily. Again, they have cell-to-cell adhesion, which results from the specific interactions of their immunoglobulin domains of two L1 molecules projecting from the surface of the neighboring cell. Each of this L1 molecule contains a small cytoplasmic domain, a transmembrane segment, several segments that resemble one type of module found in the fibronectin earlier, and 6 immunoglobulin domains situated at the N-terminal portion of this molecule. So the inset here is showing you the structure of the two N-terminal Ig domains of VCAM, an immunoglobulin SF molecule on the surface of the endothelial cells. The Ig domains of the VCAM, VCAM, and L1 have a similar 3D structure consisting of two sheets packed face to face. We can also see here the integrin IGSF interactions. The IGSF family members can also bind to the integrins of their neighboring cells. Moving forward to the cadherins. Cadherins are part of the glycoprotein family. Typically, they join cells of similar type to one another. They are calcium, on the other hand, dependent. Possibly the single most important factor in molding cells into cohesive tissues in the embryo and holding them together in the adult. Cadherin loss are associated with malignancy. So these are the cadherin that form the tip links of a cilia. Okay, cadherins are 100. There are 100 of them that have been identified in humans. The Usher syndrome is one of good example of characterization of deafness and gradual loss of vision that can result from mutations of your non-classical cadherins. So this is an intercellular junctional complex, showing you a schematic diagram of the junctional complex on the lateral surface of of your simple columnar epithelial cells, which consisting of a tight junction. Okay, this tight junction are what we call as the zonula occludens, and the adherens junction, the zonula adherens, and the desmosome, the macula adherens. Other desmosomes and gulf junctions are located deeper along the lateral surface of the cells. We can see here that there are adherence junctions and tight junctions that encircle the cell, whereas these desmosomes and gap junctions are restricted to a particular site between the adjacent cells. Hemidesmosomes are shown at the basal cell surface. On your right is an electron micrograph of a junctional complex between two rat airway epithelial cells. There are acronyms shown here. The TJ stands for tight junction, AJ stands for adherence junction, and the D is for the desmosomes. Again, cadherins are also found in these specialized intercellular junctions. They're calcium dependent, and their networks provide intermediate filaments in structural continuity and tensile strength. Now is the tight junctions and sealing their extracellular space. Tight junctions occur between the neighboring epithelial cells, showing you the figure of an electron micrograph of a section through an apical region of adjoining epithelial cells, showing here where the plasma membranes of the two cells come together at their intermittent points within the tight junction. In the inset, it will show you the tight junction structure at the higher magnification. Tight junctions block the diffusion of solutes through the paracellular pathway between cells. Below is a model of a tight junction showing the intermittent points of contact between integral proteins and two opposing membranes. Each of these contact sites extends as a paired row of proteins within the membranes, and they form a barrier that blocks the solutes from penetrating the space between the cells. So, the points of the cell-to-cell contacts are sites where integral proteins of two adjacent membranes meet within the extracellular space. Now we have to move with the intercellular communication. We have the gulf junctions. Plasma membranes of a gulf junction contains channels that connect the cytoplasm of one cell with the cytoplasm of the adjoining cell, showing you an electron micrograph section through a gulf junction perpendicular to the plane of the two adjacent membrane. We have seen here the pipelines. Okay, it resembles like a pipeline between the two cells that are seen as electron dense beads on their opposed plasma membranes. So, gap junctions, again, remember, are the sites between animal cells that are specialized for intercellular communication. These cell membranes come very close to one another, but they do not make direct contact at the gap junctions. This is a schematic model of a gap junction showing you the arrangement of 6 connexin subunits to form a connexon. They are containing half of the channel that connects the cytoplasm of two adjoining cells. Each connexin subunit is an integral protein, and they have 4 transmembrane domains. Okay, so you will show it in the figure below. Next is your experimental pathways. What are the role of these gap junctions in intercellular communication? A small subthreshold depolarization induced in the presynaptic nerve cell will produce a very rapid depolarization in the postsynaptic cell. Remember, the synapses communicate to one another, okay, transmitting action potentials. Not possible through a chemical synapse or a neurotransmitter release. It was predicted that nerve cells were connected by an electrotonic synapse, in which ionic currents in the presynaptic cell could flow directly into the postsynaptic cell on the other side of the synapse. And plant cells are separated from one another by a cell wall, and they lack the specialized junctions which are found commonly in animal tissues. And for cell-to-cell communication, most plant cells are connected by plasmodesmata. These are cytoplasmic channels that pass through the cell walls of their adjacent cells. Showing you the figure of an electron micrograph of a section through a plasmodesmata of a fern gametophyte. The desmotubules is seen to consist of a membrane that is continuous with the ER of the cytoplasm on both sides of the plasma membrane. And letter B is a schematic drawing of a plasmodesmata. The black arrows indicate the pathways taken by the molecules as they pass through the annulus from cell to cell. Plasmodesmata allows larger molecules to pass since the pore is capable of dilation. Plant cells produce their own movement through proteins that regulate the flow of the proteins and RNAs from cell to cell. This is an example of the movement of proteins from one cell to another within a plant root. The smaller inset showing you here the localization of the fluorescently labeled messenger RNA molecules in green that encode a protein called SHR. The mRNA is localized within the cells of the stele or the STE, which is thus the tissue in which this protein is being synthesized. The larger photo shown here shows the localization of the fluorescently labeled SHR protein, also the green color, okay, present both within the stele cells where it is synthesized and the adjoining endodermal cells, the END, into which it has passed away of the connecting plasmodesmata. The transported proteins is localized within the nuclei of the endodermal cells where it acts as a transcription factor. Cells of nearly all organisms other than animals are enclosed in an unproductive outer envelope. In plants, this provide individual cell support and forming the plant skeleton. Plant cell walls contain a fibrous element embedded in a non-fibrous gel-like matrix. Cellulose provide the fibrous component of the cell wall, and the protein and pectin providing the matrix. Cellulose molecules are organized into rod-like microfibrils. This is your plant cell wall. Showing you the top photo is the electron micrograph of a plant cell surrounded by its cell wall. Your middle lamella is a pectin containing layer situated between adjacent cell walls. Electron micrograph showing you the cellulose microfibrils and hemicellulose cross links of an onion cell wall after extraction of the non-fibrous pectin polymer. So the photo below is also a schematic diagram model of a generalized plant cell wall. The matrix of the cell wall is composed of three types of macromolecules. The first is hemicelluloses. These are branched sugars, polysaccharides, whose backbone consists of one sugar, example glucose, and side chains of other sugars such as xylose. Pectins, negatively charged polysaccharides containing galacturonic acid, and there are also proteins which mediate dynamic activities. So this is a freeze fracture replica of the membrane of an algal cell. The rosettes shown here are thought to represent the cellulose synthesizing enzyme or the cellulose synthase situated within the plasma membrane. Cell walls arise as a thin plate that forms between the plasma membranes of newly formed daughter cells following cell division, and the cell wall of a young undifferentiated plant cell must be able to grow in conjunction where the growth of the cell it surrounds. We have also lignin, a protein, in the walls of these water-conducting cells of the xylem by which it provides the support required to move the water through the plant against gravity. So, I hope that you've learned something today. Thank you so much for listening, and this is the Cell and Molecular Biology series, the CellVerse lining. Have a nice day. Bye-bye.